Hey everybody, just want to start jumping into the second set of notes for Mendelian Genetics here. So how are traits expressed and how are inherited and expressed. So we're going to get into some more of that. So we'll talk about linked genes a little bit today. So before we get into that, Thomas Hunt Morgan was a big geneticist and worked with a lot of fruit flies and started off a lot of research with fruit flies and gave us a lot of this information. So give a little credit to him. 1800s, Columbia University. Determined genes were carried on chromosomes, won the Nobel Prize for that. And he used fruit flies, which we talked about the other day, are good model organisms. So go back and think about what traits make a good model organism, if you don't remember. And Drosophila melanogaster, that is the genus species name for the fruit fly. So when you think about sex chromosomes, not all organisms have the same setup as far as chromosomes work. So why do males exhibit traits that are sex-linked at a higher frequency than females? So to answer that question first, and that would be on a normal system like ours, where we have the females with the two X's and the males with the X and the Y. It's really a couple of reasons, right? So because the males have two different sex chromosomes, they only have the one X, so they either have it or they don't, right? They're not a carrier. So it's a little bit higher frequency that way because they can't have any heterozygotes. So if they even get one of the alleles, then they have that disorder or that phenotype, if you will. So and then if we look at some of these other sex determination systems down here, it's pretty interesting. Let me show you a couple of these. So we have the XY system, right? So you guys should know the XY system by now, where you have the males and the male with the XY, the female with the XX. Over here, the ZW system, it's literally the exact opposite. You have the female with the Z and the W, and the male with two Zs. And so it works like our system, but male and female are flip-flop. So they just switch the Z and the W letters just so it's not so confusing. Down here with grasshoppers, we have what's called the XO system. And really, they just have one type of sex chromosome, and it matters how many of them you have. You either have one or two. Okay, so the male has one and the female has two. And then down here, I need to kind of adjust my screen so I can read it. Down here, the haplodiploid sex determination in bees. This one's a little funky. So if you look, it says the queen is diploid, right? So two copies, and the drone is haploid. So the queen mates with a drone that's haploid, and so if that happens, then you're going to get diploid workers right here, right? Because you're going to have these two parents. You're going to have these two parents contributing, and so you're going to be diploid, right? But the queen can also sometimes just have unfertilized eggs turn into drones, which are then haploid. And so in this case, you can have sister workers that are 75% related to each other. And so it's a real different type of system when you think about it, where if the if the drones do fertilize the queen's eggs, then you get a diploid offspring, and if they don't, they can still have a haploid offspring. So pretty crazy system. So a little bit more on linked genes here. So the white eye mutation is on the fruit fly X chromosome, and this is just kind of a normal result, what we would typically see. If you would have a red eye and a white eye, and then if you get the all reds, and then you do the self-fertilization or self-crossing, or cross them with like ones, then you're going to get this 3-to-1 ratio, right? 3 reds to the 1 white on the F2 generation, and that's typical, right? We've seen that ratio happen a couple of times. We looked at that with Mendel's fruit pea plants. So if we go on, though, one of the points is when you have these linked genes, they can give you different combinations. They do not always follow. So the typical 3-to-1, when we get that, the typical 3-to-1 ratio does not always happen when you have sex-linked transmissions. It can happen in different combinations. So if you see combinations in monohybrids, they have to be in the the linked genes. They have to tell you in the problem that the genes are linked, or they might just say sex linked, right? Because we can tell down here with the X and the Y's that are in the problem that this would be a sex linked issue, but you don't always see the 3-to-1 type patterns down here. So just be aware that when you have linked genes or when you have sex linked genes, you can have different ratios than what you're used to seeing. So, and that's because of things are going to donate things at different frequencies because the males don't have the 2 to donate, they only have the 1. So that's going to change the ratios that you'll see the outcomes in. Okay. So linked genes usually travel together, and it says why? Well, typically they're pretty close on the chromosome. So, if you have chromosomes here, I always forget it changes the color. If two genes are really close together, even if you have crossing over from a section, and this is going to cross over and leave, most of the time your two linked genes will travel together because they're close on the same chromosome. When might they not travel together? If they're just far enough apart to where, maybe they're like this, but crossing over happened right in between them, and this top piece is going to cross over with a different chromosome, but this one stays, right? So then at that point, they might not travel together. Excuse me. What correlation can be drawn from this? It's really just that the more recombinants you get, the more crossing over you're going to have. And so the increased frequency of crossing over is going to kind of show you how much of this is going to happen. So remember when we talked about map units and we talked about the distance between the centromere and the genes, and the farther they are from the centromere, the more likely they are to cross over, right? And then that crossing over, the more often that happens, the more recombinants we get. And remember, recombinants are things different from the parents. And so we get more variation this way, okay? So here's a little thing about map units. And you can dig into the picture a little bit. You can pause it and dig into the picture and the numbers here, but I'm just going to focus on, for now, just this concept right here, that one map unit is 1% chance of crossing over or having recombinant offspring, right? So, if you look at the parents and then what offsprings they have, gray body, straight wings, black body, curved wings. So if you look, these offspring would be the same, but these over here would be your recombinants, right? These are going to be the recombinants. So the higher percentage recombinants you get, the more likely it is that crossing over took place and we got some sort of variation there. Okay, so pretty straightforward correlation. So again, just the same kind of thing. It just shows you the distance on the chromosome, and the point is that it's more likely that these would stick together, right? That you're going to see a black body and cinnabar eyes. It's more likely those are going to travel together because they're so close. When you have crossing over, something like this and this way down here, it's more likely that the crossing over would have happened between them and split them. Okay. Oops. So now we're going to get into a few other things here, a few kind of miscellaneous type genetic things. So non-Mendelian inheritance, the title of this slide. So things that don't follow Mendelian patterns. We have things that are extranuclear genes, so genes that are outside of the nucleus. And remember, mitochondria and chloroplasts have their own DNA. They have chromosomes. And these get passed on matrilineally or through the mother. And so that's not Mendelian, right? This is a different way to inherit things where only the mother's DNA would matter because they come through these particular organelles, the mitochondria and chloroplast. So variegated leaves in hostas and things like this are passed on by the chloroplast gene. And so that's passed on matrilineally or through the mother. We also have things that are passed on patrilineally. And the Y chromosome is passed on patrilineally. Now, it's not extranuclear, but it is non-Mendelian. Okay, so it's still within the nucleus, right? Because it's a sex chromosome that's going to be in the nucleus, but it's not going to follow Mendelian patterns because it's just guys donating it, right? Mendelian patterns have to deal with the two parents. So it's pretty interesting, though, that they actually use this. They tried to track basically history of man and where they migrated over thousands of years based on the evidence they found in all of the Y chromosomes. and the mutations of them, and it helps them understand these human historical migration patterns all over Europe. So pretty cool that they can trace that stuff back through history. Epigenetics is not Mendelian, okay? Epigenetics means outside genetics. So this is actually some differences that can happen not really because of any particular allele or order of bases, but for other reasons. So genomic imprinting is one, and this is where it only matters if the trait's expressed in one parent or the other. And so let me grab my pen again. So over here, so the blue one is going to be the father's chromosome. The red one's going to be the mother's chromosome. And so the normal allele for the father is expressed. The normal allele for the mom is not expressed. And we got a normal mouse, okay, or the wild type, the regular version, right? So if we go down here and it says mutant version of the allele inherited from the mother. So down here, this one right here is a mutant, meaning a mutation. But the father's is still normal. So what happens to the mouse if we have a mutant mother allele and a normal father allele? Regular mouse, the same as this guy right here, because it's only mattering what the father's chromosome is in this case. So here we got a normal from the mother, but we got a mutant from the father. Okay, but it does matter what the father's is. So now the mutant is being expressed because it was on the father's chromosome. In this case, it could be, for a different example, it could be the mother's, but for this case, it's the father's. So now we get the mutant dwarf mouse, right? We get a smaller mouse because it was expressed on the one that mattered, and the other one really wasn't playing a role, right? And that's non-Mendelian. That wouldn't make sense for a Mendelian type pattern. Another non-Mendelian is methylation. This is also epigenetics. Methyl groups are added to DNA to inactivate the DNA. So if we look down here, it says there's some evidence that suggests methylation of DNA is heritable. This could explain imprinting. So we have unmethylated DNA down here, which is the blank circle, and we have methylated, that is the gray circles, which are these right here. So basically right in front of the gene, we have this section of DNA that's going to impact whether there's going to be gene expression or not, okay? And in this case, we have unmethylated DNA, so we're going to get gene expression. And remember, gene expression just means that the DNA was converted into RNA, and that RNA was converted into a protein, and that protein is out there doing the job that the gene gave it the instructions for. So the gene has the instructions, the protein's going to go do the job of those instructions. So the gene is being expressed by the protein doing the work. Okay. Down here, when you have methylated DNA, what happens is methylated DNA, instead of being accessible and open, it becomes closed and all kind of balled up. And then there can't be transcription and there can't be translation. And so you don't get any gene expression. You can't make the protein when it's all kind of curled up and no other proteins can get in there to do their job. No RNA can be made when it's methylated. Okay. So things like this, like methylation, would fall into these categories of influencing the genetics under non-Mendelian patterns. Environmental influence also, there's environmental factors that can affect genes and can affect how they are expressed. And soil pH, on this example, soil pH can dictate a change in expression. The more acidic the soil, the bluer the flowers. Coat color can change for rabbits and based on seasonal basis. So definitely environmental factors can play a role, which is kind of leading us into just one slide here on, I like to quote up there, how much of a human is a product of its genes. That's something people have been, kind of conversing about and talking about for a long time is, are we just what our genes make us, or does the environment play a role? And I think it's pretty clear to most people that, your genetics lay the foundation, but what environment you are brought up in or what environment plants are around in or whatever can certainly influence the outcomes of genetics and of the phenotypes that we exhibit. So definitely plays a role. Pedigrees, we're going to move into some pedigree stuff here. I'm going to give you a little sample here, but you're going to do, or maybe you already did, some pedigree problems that I posted, which is really just family trees, right? Pedigrees are really just family trees. They're kind of old school family trees. So when you draw one, you just pair up the spouses and then, drop a line down and then spread out their kids. And males are always drawn with squares. Females are always drawn with circles. If they're colored in, that means they have the phenotype, like hemophilia or whatever disorder you're looking at. If they're colored in, that means they have it. If they're blank, that means they don't have it. And this one doesn't show it, but I can draw one in here. If somebody had half a box, okay, which I kind of drew outside the lines there, but if somebody had half a box where it was half shaded and half not, what do you think that would mean? That would really indicate that they're a carrier, that they don't have it, but they still could pass it on. So typically those are heterozygous, right? Where they don't display the disorder, but they could still pass on one of those alleles to give the offspring the disorder. So that can happen as well. Mostly modern genetic testing can tell us genotype directly now. We don't really have to do too many pedigrees by hand, but we do them by the computer a lot. And the idea behind them is still the same. And we don't have really fancy programs to be doing pedigrees, so we still kind of do them paper pencils. That's how that goes. So a few points to keep in mind. There are very few traits that are inherited in strictly Mendelian patterns. So all those Punnett square type things we do, that's really very minimal. Most genes do not follow Mendelian patterns. There's only a few that actually follow that. So just keep that in mind. From an evolutionary perspective, there are traits that can confer fitness in one circumstance, but could be very detrimental in others. And you might be able to tell that from this picture right here, kind of comical little picture here. But another example would be like, the frog with the best mating call, right? Like that's really good in that circumstance because you'll get mates. But you're also giving away your position, and your predators might find you more easily, which would not be good in that circumstance. So that's kind of what they're talking about, where sometimes you can maybe have too much of a good thing. Okay, so we are going to run through a bunch of disorders here, just to kind of cursory look at how are these disorders passed on, and not all of them are actually disorders. This first one is not a disorder. It's just looking at widow's peak, which is the little pointy part you get in your hairline if you have that. So widow's peak is an autosomal, which remember, autosomal means the chromosomes 1 through 22, so not sex chromosomes. Okay, so it's somewhere on one of the normal chromosomes, and it's dominant. And so autosomal dominant. So you have widow's peak, which would be big W, or big W, little w. So as long as you have one big letter, you can get a widow's peak. You have to be totally recessive in order to not have a widow's peak. If we also, an autosomal dominant one is achondroplasia. This is where the cartilage does not form properly as arms and legs grow. So you pretty much have a normal sized trunk and head, but you have really short limbs. And so the homozygous form of this is lethal, and you don't have people that exhibit this genotype because they would not make it. But if you're heterozygous, then you might have achondroplasia. And I put Peter Dinklage in here, who's a pretty famous achondroplasian that has made a really good life in movies and stuff. He's also the elf, or not the elf. He's the businessman in Elf in the movie that runs down the table and starts the fight with the elf. So, two pictures. So, acromegaly. This is a form of gigantism, so also autosomal dominant. So an overactive pituitary gives you lots of growth hormone, and you become really big. And this is Andre the Giant. He's a former WWF wrestler, like the wrestling you would see on TV. I think he was about 7'6", if I remember correctly, and probably 400 pounds. And he doesn't look like he's actually a big blob because he's so tall. But you also get this really kind of long, heavy face and overgrown features and hands that are just huge. Huntington's disease, you get many CAG repeats, and those are cytosine, adenine, guanine, like DNA bases is what that means, where they repeat, and it causes a lot of problems. Oops, so sorry. And so this leads to degeneration of your nervous system. which is really difficult and sometimes painful, and you also lose the ability to kind of control your body. So it's not a very fun one at all. Woody Guthrie, famous musician, had it. 13, I put in there. She's just a television character on House, but she had Huntington's in the show. Autosomal recessive. So attached earlobes show a recessive inheritance pattern. So um, if you have attached earlobes, then that would mean you are um, little f or homozygous recessive. And you can see like people that have a free lobe versus people that kind of their ear just goes right into the side of their head. And so that's an autosomal recessive. Another autosomal recessive is albinism. So the mutation in the melanin pathway where melanin is not produced. And so, it's not really known, actually, not really thought about a lot, but, humans have albinos, but there's other organisms that have albinos as well. I thought these were some pretty cool ones, so threw them in there. PKU, phenylketonuria, also known as PKU. It's a dangerous one. You got to watch out for this one if you have it. This is one of the things, I don't know if you guys know, but right when a baby's born, they generally do a few quick tests on that baby so they know, especially if there's like food allergy type things, they do those tests right away. And this is one of them. So they usually like take the baby and they like put a prick in its heel and take a little drop of blood and then do a quick genetic test to see if they have PKU. And so if they do, then they cannot digest phenylalanine, which is an amino acid that goes into proteins, which is in a lot of foods. And your body, if your body can't break down phenylalanine, then it accumulates in your brain and it will lead to mental conditions. And so it can be treated through diet. It is required by law that if your food has phenylalanine in it, you have to put it on the food label. And it's right there on that yogurt container. So anyone that has PKU can never take in phenylalanine. You have to avoid that at all costs. Cystic fibrosis, you guys may have heard of this one before. It's a defect in the chlorine ion channel, leads to a buildup of thick mucus in your respiratory system. It really is a tough one. It can be really difficult, and a lot of people don't live longer than age 50 if they have CF. So the kid on the right is wearing a vest that vibrates him, so it hopefully breaks up all the mucus, and then he can like get it out, or it goes in or out of his system one way or the other. And so the guy on the left is also trying to break up the mucus with I guess massage or aggressive massage, trying to manage those symptoms. It's not a fun one at all. X-linked recessive disorders, most common sex-linked disorders. These are like the ones you've been doing in your Punnett square problems. They show up in males most often due to hemizygosity. So hemizygosity, it just means that you only have one copy. And so males only have one X chromosome, so they only have one copy. And thus, because they either have the disease or they don't, and there's no heterozygous version, then they show up at a higher frequency with the disorders. Muscular dystrophy is this way. So abnormality in the dystrophin protein, and you get early onset muscle degeneration. Unfortunately, symptoms by age 6 and death by age 20 is fairly common. And so, again, not a very good one at all. Hemophilia, this was one that you guys had in your problems. So this is where you can't clot the blood, and it could lead to death by hemorrhage, meaning you just bleed out because your body or your blood won't clot fast enough if you have a big enough injury. Can be a difficult one, but now we can treat it a little better. People have preventative clotting factors that they take as medicine to kind of help them a little bit. This was just a pedigree of a former royal family here. If you look, Victoria, Queen of Great Britain, and Prince Albert, and this is their family, and had quite a few people with hemophilia in their pedigree or in their family tree. And a lot of that is because, back in those times, a lot of royal blood would mate within their bloodline because they didn't want to mate with commoners. But then you couldn't ever breed out the hemophilia. You could never breed out the disorder. It just kept showing up again because they were mating with people that were a little too close in their family tree. Another X-linked recessive, red-green color blindness. So defects in proteins that detect the wavelengths of red and green light, harmless unless you want to be a pilot. But I don't know if you can see the numbers that are in these circles. If you can't see that there are 2 numbers in those circles, then you might have a little bit of color blindness. You should ask your eye doctor about that next time you are in there. But you should see 2-digit numbers in each one of those. X-linked dominant, not very common, but you can pick them out on pedigrees pretty easily. If you have a few generations there, you should be able to tell, but they're just not very common that you have an X-linked dominant one. Y-linked dominant, even less common, mainly just because there's only a few genes on the Y chromosome. Yeah, guys, our chromosome just doesn't have a lot of value to it other than to make us guys. That's really about it. And that's all I got for you. So hopefully you learned a little bit about some genetic disorders and how those go on, and hopefully that was helpful to you. We'll talk to you soon.